Antibiotics are the drugs that are active against microorganisms, particularly bacteria. As you know, bacteria are two types, gram-positive and gram-negative. Here, antibiotics general classification, we first introduce beta-lactam antibiotics that contain lactam ring inside, beta-lactam ring inside, and second, aminoglycosides that contain glycosides in their structures. Macrolide antibiotics, there is macrolide ring in this, and tetracyclines contain 4 ring in their structures, and fluoroquinolines contain fluoroquinoline ring in their structures. Generally, first thing I want to clear to the students, maximum students thinks that the antibiotics are also synthesized in the laboratory, but maximum antibiotics are not synthesized. These are directly obtained from the maximum fungal species through extraction, and some are semi-synthesized, and fluoroquinolones are the synthetic antibiotics. So it should be clear in the students. So here we talk about the beta-lactam antibiotics. You know, beta-lactam antibiotics contain a beta-lactam cyclic amide 4-membered ring inside the structure and one 5-membered ring thiazolidine. So here CIFM, that is known as CIFOM, and there are two major category in beta-lactam ring. That first is penicillin, that is very popular, and second cephalosporins that are more popular. Penicillin was discovered by Alexander Fleming by chance discovery. That is very interesting. He was working on Staphylococcus aureus. Culture was grown in the microbiology laboratory, and he went to the summer vacation. As he returned back from the summer vacation, he noted down in the petri dish of Staphylococcus aureus, there was some secretion of fluid that was secreted from a fungal species known as Penicillium chrysogenum, and there was no growth of Staphylococcus aureus in the region. There was liquid in the petri dish. So he isolated the liquid from the fungus Penicillium chrysogenus and used against the microorganism, particularly gram-positive bacteria, and this fluid inhibit the growth of the microorganism Staphylococcus aureus, and we have Streptococcus pneumonia-like microorganism. This is the discovery of penicillin. Then Florian Chen and Fleming prepared a slant of this fungus Penicillium chrysogenus, and they fermented in big fermentator, and after optimizing the condition pH and nutrients and temperature, then after maximum growth he collected the liquid and formulated the penicillin as a injection and some tablets after that semi-synthetic procedure. This is the method of penicillin, and this was first used against the mice that was infected from streptococcal bacteria, and 3 injections were given to the mice, and mice was recovered from the bacterial streptococcal infection. Then it was used against the human being in Second World War, and there was a recovery from this antibiotics. But what was the major drawback of this penicillin? This is liable to beta-lactamase and acid liable, means can't take as a tablet, degraded by HCl and inactivated by beta-lactamase. So there are some modifications have been done in this, and extended spectrum like penicillin G is well known. Then beta-lactamase resistance bacteria are found, and the antibiotics also developed, beta lactamase inhibitor, and some acid labile also prepared, like cloxacillin, ocloxacillin, dicloxacillins, and some penicillin, like you can say ampicillin, amoxicillin, also developed. And there is combination of both drug cloxacillin and ampicillin was given. That was perfect. And amoxicillin with clavulanic acid, that was beta lactamase inhibitor. And this augmentin was more effective as compared to the simple antibiotics. And then cephalosporins are developed first generation, second generation, third generation, and fourth generation, that are more effective. These are especially effective against gram-positive bacteria because muramic acid and some sugars, aminoglycosides are in their structures, so they mainly inhibit cell wall synthesis, and their allergic reaction also with penicillin. Then first generation cifloxime is well popular. Second, ciflor, third generation, or parenteral antibiotics like cefotaxime, ceftriaxone, and orally cefixime is more popular. Now, aminoglycosides are effective against gram-negative bacteria. First was the streptomycin, that was isolated from Streptomyces gaseous, and then the gentamicin and kanamycin and amikacin. These are the aminoglycosides and more powerful. These are not absorbed from the GIT, so these are currently used injection form. And some kanamycin and others are also used in topical application. And then macrolides are effective against gram-positive bacteria, alternate to penicillins like erythromycin, doxythromycin, and azithromycin is more popular due to high penetration and effective in gram-positive and complicated infections. Then we talk about the cephalosporins. Cephalosporins are the synthetic antibiotics particularly effective against gram-negative. First one, like ciprofloxacin or floxacin, ofloxacin, and genfloxacin, then modified with extended spectrum moxifloxacins that are effective against gram-positive and gram-negative bacteria. These are first effective against Escherichia coli, that is opportunistic bacteria convert into pathogen and during immunocompromised patient. And then tetracyclines, chloramphenicol are the known as broad spectrum antibiotics, and these are used particularly against unknown disease like spreading cholera, plague-like conditions. We can use, these are more popular. And that oxytetracycline, minocycline, doxycyclines are the more powerful. And their major problems are their inactivation, some enzymes that are produced by the bacteria. So enzyme resistance antibiotics are more produced at present, and this is the major scenario at the present world. There is a lot of resistance to the uh, antibiotic to the microorganism that create problems in the treatment. So first is the selection of antibiotics to the particular disease. Like there are many causative organisms that are effective to the particular antibiotics. Escherichia coli gram-negative effective against fluoroquinolones. And gram-positive bacteria, penicillins are very effective and alternate if allergic patient, then you can use macrolides like erythromycin and doxythromycin and azithromycin. Now, some unknown where disease are there, you can go for broader spectrum antibiotics. And like some non, you can say here typhoid is here Salmonella typhi, you can treat with antibiotics that is effective against gram-negative, and some Corynebacterium diphtheria against diphtheria gram-positive, and microorganism Mycobacterial tubercle is atypical microorganism bacteria, that is acid-fast bacteria, and we can treat with some special antibiotics.